In this video, we will show how to speed up HDX analysis by using single sample experiment mode. Conventional experiments in HDX, such as a protein with many ligand-bound states, would require reanalysis of the apo state. Here, we will show how to analyze everything as individual sample experiments, create any comparison, view the results using multiple tools, and additional automation features will be shown. So we'll just create a new project, give it a name, single sample project, and select a location. And then, assuming we have our protein previously set up, we will select the project and just set it up using the single sample experiment wizard. So we have one protein, 4 experiment samples, which in this case will be apo versus 3 ligands, 5 time points, 2 replicates, and we'll put in the additional 0.8 and 0.7 for deuterium solution and recovery. Optionally, one can add experiment information in the text box below, including custom information. So we'll select next, select the previously set up protein, and we'll just give each one of the 4 samples a name. Apo, ligand 1, ligand 2, and ligand 3. We'll ignore the prefix fields for now. The first time point always has to be 0. It is in units of seconds. So we have 0, 10, 60, 900, and 3600 seconds. Select the drag to drag the files in. And then we'll just start dragging the individual files in. So this does take a bit of time, but we are also setting up 4 individual single sample experiments in one go using this single sample experiment wizard. So when this is completed, we will have 4 experiments set up. Let me just resize this. Okay, so ligand 1. So this data set is a good example data set. We only have 2 replicates, but for illustration purposes, we will show you. So there I put deliberately a file in the wrong slot so that I can show you that it can be corrected by dragging the correct one into the right spot, and it'll just update the file path. It's not actually copying the files yet. (901), 2. Okay, so 3 samples have been completed. Now we're looking for the 4th, which is ligand 3. Almost there. Okay, so when you're finished with this, select the next button, at which point the software actually creates the folders for each individual experiment and actually does the copying of all of the files. It'll tell you that the setup is complete, and then you have to go to each individual experiment. So what we'll do is right now we will go to we can see that everything's set up correctly, and we'll just go to our apo, and initially we will fire off a T0 detect. So this will just search your existing peptide set in the undue-derivative controls. We've covered the parameters in other videos, so I won't get into this at this time. Just change a few things based on known instrument settings. And I'll select Run. So right now the software is setting things up and searching for your peptides in the undue-derivative control. In this case it's only one file, so it really doesn't take long. And then we can go into the Jobs Run folder. There is a job named as we named it. Select View Peptides. I'll go ahead and resize the window so that we can get a better view. Okay, so initially we will go back into the APO dataset, and let's look at the results of the T0 search. So we have our grid. We could resize the pane, look at the result spectra, zoom in, zoom out, look at our extracted ion chromatogram. You could scroll through peptides row by row. At this point, once again, we're just trying to establish whether the identifications are correct. If there are peptides that are not identified in the 0 seconds, there's no point in pursuing them in the on-exchange data. So I'll do a quick initial scroll. There's an overlapping peptide which does not affect the result. Theoretical distribution. Okay, so these appear to be pretty good. We'll just select all of them. Save the selected peptide set. Give it a name. I'll give it working. And then go into the peptide set. Select that exact working peptide set for HDX detection. So now when I select Run, it will search the peptides that I selected in all of the on-exchange raw files. So this will take more time than just searching in the undue-derivative controls, obviously. There is a status bar that shows you various things such as memory allocation, how many jobs are being processed, and elapsed and estimate for time remaining. So at this point, I'm not going to cut the video because I want to give you a sense of how fast the analysis can take. Okay, and it's done. So now we can go to the job, select perturbation view, and look at the HDX results for this single sample experiment. So there's the 0 seconds spectra. We could select various individual spectra by selecting these cells, or multiple at the same time. It looks like the software did a reasonable job finding all of those. And then we can click through each peptide, change the retention time or the MZ limits as is described in other videos. So here we're setting the MZN for all of these on exchange. It does a recalculation. It generates the average mass spectrum for all of them. This is the extracted ion chromatogram where the software has found the peptide. Okay, we'll do a quick few checks. These peptide results seem fairly reasonable. Again, most of these features are described in other videos. Let's select, zoom in on the spectra, look at the theoretical isotopic distribution overlay. Okay, so at this point, I would say that the data is reasonable. So if we want to run an HDX on all the next 3 ligands, we could select this HDX all and then just simply browse to the working peptide set that we had created in the previous one. This can save quite a bit of time. So we'll just say, let's reuse that. Select your parameters and then press the run button. So a key time saver here again is that we're skipping the entire T0 detection step for all of the subsequent ligand experiments. In addition, we're running the HDX detect reusing the peptide set, the working peptide set from the APO experiment. And the software is going ahead and running all of the HDXs in one go. This process is fully automated. We'll just go ahead and skip ahead. Okay, so all of the detect jobs are now completed. So the next step is to go into each individual HDX detect. There will be a job called auto HDX detect in each. And we could load those into the perturbation view. Go ahead and use all of the tools that are available to look at the data. So here we could look at the consolidated results for the single sample. Let's look at the perturbation for ligand 1. And essentially you go through your data set as you normally would. So once you're satisfied with the results, then we set up our comparisons by selecting the experiment comparison setup, which launches this view, and it allows you to select add comparisons using the add comparison button. In this case, we'll go APO versus ligand 1, APO versus ligand 2, and APO versus ligand 3. There is only one protein available, so we don't have to select that. And we can also use these text box at the end to provide an alias or a synopsized name, which will populate the column header. We can go back in there and make sure everything is saved correctly. Everything looks good. We make edits if needed. Save and close. And now essentially the software knows which single sample experiments to compare based on the information provided here. So now we could just select experiment comparison from the project menu. And then it's telling us that all of the data needs to be finalized. So we need to go back into each individual experiment. This is not a difficult task. And just select the finalized data. We do this to lock in the results and to make the data available for the subsequent experiment comparison view. So we'll just finalize the data from that. The software also exports all the plots and does a full CSV export at the same time. And then the final ligand 3 experiments. Okay, now that all the now we should just be able to run the experiment comparison view, and it loads the results into a nice grid here. We can add the color heat map. We also have the ability to run the numbers on just the first sample or the second sample. You could select a subset of time points. On individual samples, a single time point, usually we'll just go ahead and select all. The remove color button will allow us to remove the colors. Now that the colors are removed, we also can delete rows. So let's select that row. We delete it, and then we could visualize that again. There's this select button that allows you to select either an individual cell, a column, or a row. So these are selection modes, and it's important to understand. It allows you to select various things. So if we select an individual cell and then hit view plots, we can see the plots that pertain to that particular peptide in both sample states. The usual zoom in, zoom out graphic export features are available. Deuterons on the right side. One could select two separate cells, visualize the plots. So there is some flexibility here. The next is when you select the export CSV, it will export this grid. Just give it a CSV name, and then when you look at the export, the actual, there's additional significant digits if you'd like to use that to plot bar charts or what have you. If you select column select, then you could select sequence coverage. It will show you all the peptides pertaining to that. You have the usual features: consolidated track, change the width, font size. Select a different column. We'll show those peptides, sequence coverage heat map, or whatever differential experiment you'd like to visualize. Then we have this consolidated view, which takes the results of the single amide resolution algorithm and maps them onto an XY block chart. It allows you to zoom in and zoom out, resize, and create whatever size chart you want, zoomed into whatever range of data is preferred. If the columns are moved, they are moved accordingly in the residue consolidation view. And then we also have the PyMol differential, which requires you to select a column. So let's take off the color. Column select. This will only work if a PDB file is available in one of the protein definitions. So this looks interesting. Let's compare it with the grid as well as the sequence coverage view with consolidated residues. Add column select, select a column, go back to consolidated view. Okay, so let's compare these results. The results in the PyMol overlay. And we could see that they line up perfectly. The colors, again, in all of these views should be identical. The next feature that will be illustrated if you plot a single cell and view the deuterium uptake plots, one can select this Prism clipboard button, which will copy the individual results in text format and allow you to paste it into Prism, which I don't have available on this computer, but essentially if you set up a standard XY Prism plot, this will paste in directly and allow you a quick way to generate publication quality uptake plots. The last thing I'd like to show you is, now that we've completed all of this analysis, how do we add additional ligands to the existing experiment? So if we select the experiment single sample experiment wizard, we could select the same project this time with just one sample. It could be a large number of samples. The same time points, 2 replicates, the same values, but this time we'll add in ligand 4. So we're creating a new experiment with a 4th ligand. Select the same sample. We'll just call this ligand 4. Add the time point information and select next, drag, and then we'll drag our files over. Okay, we'll select next. Software will copy, create the experiment. Here's another new feature it's called HDX detect from an existing experiment. So the way this works is one can right click on the protein node and select HDX peptide set from a previous experiment. In this case, we'll select that same working peptide set as we had previously. It copies it over and allows us to go straight into running the HDX detect. Again, saving time. By bypassing the T0 detect. Okay, the detect has completed. Now we can go into the perturbation view, view the results, curate the data, examine the peptides, and essentially do our usual data curation process. Everything looks good. You could also double click on a peptide here, it'll automatically be selected in the grid. Alright, let's finalize the data, add it to our comparison. Now we'll add a new comparison, call it APO versus ligand 4. Let's give this an alias as well. And then save and close. And if we relaunch the experiment comparison, you notice there's a new column with data from this new experiment. You could select the column and view the sequence coverage here as well. Once again, this is differential sequence coverage between APO and ligand 4. In addition, it shows up in the residue consolidation view, which displays data at the single amide resolution using one of 2 published approaches. Analyzing this experiment, if done the conventional way, would have involved analysis of 8 samples. Using the single sample experiment method, only 5 samples are analyzed, resulting in a data reduction of 38%. Looking at the workflow, we've streamlined the input process by providing import tools and automating the setup of multiple experiments. We also allow the bypassing of the T0 detect for all subsequent experiments and automate the HDX on-exchange detect jobs running sequentially without user input. By combining these approaches, we can easily cut our time of analysis in half. For more information, go to hdxworkbench.com or contact us at the information provided here. Thank you.